Welcome to the MEF2C cast, a podcast dedicated to educating, supporting, and building community around MEF2C haploinsufficiency syndrome.
The MEF2 Seatcast is for informational and entertainment purposes only. Views expressed are those of the hosts and guests and not necessarily of the podcast or its sponsors. This is not medical advice. Consult a healthcare professional for medical concerns.
Alrighty, hi everybody. Welcome to the MEF2C cast. Before we send you over to our interview, I just wanted to take a minute to introduce our guest. I'd like to introduce Jamie Lundy. Jamie Lundy is a certified genetic counselor. She has a Bachelor of Science in Biology with a minor in psychology, as well as a Master's of Science in Genetic Counseling degree. Jamie works for a hospital system in Pennsylvania, where she works clinically with patients and on the research project Simons Searchlight. And now a little about Simons Searchlight. Simons Searchlight accelerates research into rare genetic neurodevelopmental disorders, including MEF2C, by collecting high-quality natural history data and building strong partnerships with families, researchers, and advocacy groups. Through online research surveys and optional blood samples, we gather detailed medical and behavioral information. We share de-identified data and samples at no cost with qualified researchers to advance discovery. Families receive data findings, help shape the research, and are empowered through shared knowledge. Simons Searchlight also manages gene-specific pages for all conditions, including MEF2C, which include gene guides, quarterly reports, and other support resources. Participation is free, and families earn gift cards for completing eligible surveys. All right, guys. And real quick, if you can, please participate in Simons Searchlight. It costs nothing but your time. You just fill out surveys and reports and things on your child with MEF2C, and that helps provide researchers with more data. And more data is one of the really important things we need right now. Dr. Chung talked about, the issue of needing to be represented so that we can stand up for ourselves and make sure that the MCHS gets the attention that it deserves from the medical community. So, as I said, it costs nothing but your time. And I mean, for Pete's sake, they give you gift cards for filling out these surveys. So, please go participate in Simons Searchlight if you can and enjoy our talk with Jamie Lundy. Thanks, everybody. Alrighty, hi everybody, and welcome to the MEF2C cast. Just Eli here tonight. Haley's a little under the weather, but I am here with Jamie Lundy, a genetic counselor that works for Simons Searchlight. How are you today, Jamie?
I'm doing very well. Thank you for inviting me today. How are you?
I'm good. Thanks so much for your time. Can you tell us a little bit about how you got involved just in genetics and Simons?
Yeah, absolutely. So, as you said, I'm a genetic counselor by training, and growing up, I always knew that I wanted to go into some type of helping career. I think I always kind of gravitated in that perspective. And just for practicality's sake, I liked the idea of going into the medical field in some capacity, but I'm very squeamish. I don't like blood or, bodily fluids or anything like that. So I found genetic counseling, which really marries genetics, but also working in a medical field. So working with families and patients. And it's really the best of both worlds. So I really enjoy the career in the time that I've been in it.
Sure. Awesome. Can you talk a little bit about your role at Simons Searchlight?
Absolutely. So I guess taking a step back, I work for a hospital system in Pennsylvania and I have a split role. So some of my time is spent working in clinic with patients. I work in pediatric neurogenetics. So I work with kids that have neurologic diagnoses, things like epilepsy or neurodevelopmental differences like autism or developmental delays. So I do that type of clinical work. And then also I work on Simons Searchlight, which is an international patient registry, and I can certainly talk more about them. But it's nice having responsibilities in both of those worlds because I think the clinical end of things makes me stronger in the research world and then vice versa.
And you spoke to wanting to help people, and those are just kind of two different, being able to help people in two different ways.
Hmm. For sure.
Yeah, that's awesome. So we are going to, before Jamie gives us kind of a crash course in genetics, we're going to roll through just sort of a quick history of not so much genetics, although it's related, but DNA in particular, because I think it's important to understand sort of how relatively new this science is. Um, So DNA was first identified by a scientist named Frederick Miescher in 1869. He called it nuclein. Um, they isolated protein in the cells and believed they transmitted hereditary traits, but they didn't understand the actual molecule itself. In 1944, a scientist named Oswald Avery identified specifically DNA as the carrier of genes. And then, of course, everybody knows in 1953, Watson Crick and unfortunately the oft forgotten Rosalind Franklin identified the double helix structure of DNA and really kind of got to the bottom of how DNA is built. From there, in 1977, Frederick Sanger developed rapid sequencing techniques. He ended up winning the Nobel in biology, I believe, for that in 1980. And a lot of those sequencing techniques we either still use today or sort of build off of. Um, Huntington disease was the first genetic disease specifically mapped to a gene in 1983. In 1990, the Human Genome Project started, finished around 2003. It sequenced about 99% of the human genome, but so 99% still left a lot to be desired. In 1993, the MEF2 gene itself was discovered. Um, in 2009, the MEF2C gene was associated with MCHS by Dr. Stuart Lipton and his team at the Burnham Institute. And then in 2022, a more correct and complete genome was published by the Telomere to Telomere group. So, all of this is to say that we are still in the very infancy of sort of the potential of genetics. There are between 4 and 500 diagnosed cases of MEF2C right now. So, I think it's important for people to understand that this is a very new science and a very new condition within that field. So a lot of questions people have, we just don't have answers to yet. And there's a lot of great people, you and others, who are working to get us answers on those, and I'm sure that they will come quickly. But in some sense, it's really exciting because we're kind of at the edge of this frontier, and there's a lot to be explored.
For sure. I think that's a very common theme in genetics that we feel like we know a lot, but then at the same time, it's really the tip of the iceberg and there's so much more underneath the surface that we don't
Yeah,
know.
right.
So,
And
very
genetics, I
interesting.
mean, kind of everything's infinitely complicated when you get down to it, but genetics in
Mhm.
particular is so insanely complex that it's really hard, I know, to give simple answers to simple questions. So, would you like to start us off with the basics of DNA, kind of the difference between chromosomes versus genes versus DNA?
Yes, absolutely. I think genetics can be like its own language at times, whether you've been in this realm for, 10 years or even 10 days. I think that we're always learning and there's even updates to terminology. So it can be helpful sometimes just to go like back to the basics, almost start over with a quick refresher. So most people are familiar that our bodies are made up of millions of cells, whether, that's a piece of our skin cell or our brain cell or our heart cell. And if you open up any one of the cells that we have, you can look at our genes and our DNA. And our genes essentially just serve as the instruction manuals for how our bodies are built, but also direct how our bodies are growing and developing over time. And everybody has about 20,000 genes in total, so 20,000 individual instructions. And because we have so many genes, they're packaged up into these larger structures that are called chromosomes. So our genes are very tightly wound up, kind of like a big ball of yarn, into these big, long structures called chromosomes. And our chromosomes come in pairs. So likewise, our genes come in pairs. And we have 23 pairs of chromosomes. They're numbered from longest to shortest. So the first set of chromosomes are the longest, and then the 22nd set of chromosomes are the shortest. The MEF2C gene is on chromosome number 5, and why that's important is because our last set of chromosomes is our sex chromosomes. So people that are assigned female at birth have 2 X chromosomes, whereas people that are assigned male at birth have 1 X chromosome and 1 Y chromosome. So if a gene is on one of the sex chromosomes, then based on someone being assigned female at birth or assigned male at birth, that can change how they may show symptoms of a genetic condition. MEF2C, because it's on the fifth chromosome, it's not influenced by biological sex. So that's just why I put that out there in case that's of interest. But if we actually zoom into the spelling of our genes are made up of what's called nucleotides. And I like to make the analogy of nucleotides are like letters. So there's four possible nucleotides that make up genes. This is A, T, C, and G. So a gene might read something like A, T, C, G, 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 T, C, something like that. And something else that's really important to understand is that our nucleotides are read in what we refer to as the triplet code. So they're read in these sets of three. So something like ATA is read in that little triplet code, and then CAG is read in that little triplet code. And each unique triplet code corresponds for a specific amino acid. And there's 20 possible amino acids. Just to give a few examples, there's alanine, valine, isoleucine. The list of 20 amino acids goes on and on. Like I said, with the triplet code corresponding for one amino acids are then strung together kind of like beads on a string to then be folded up into a much more complex protein structure. So at the end of the day, it all goes back to the nucleotide code corresponding for amino acids, then are folded into much more complex protein structures.
So those triplet codes code for amino acids, and then those amino acids assemble into a protein. So is a gene a series of triplet codes that completely make a protein? Is that the right way to think about it, or a way to think
Mhm.
about it? Okay,
Yes. Yep, I would say that that is correct. And then
gotcha.
proteins have various roles in our bodies, whether that's going to build a protein that's really important for brain function or a protein that's really important for heart function. And then some proteins have a lot of roles, so may influence the brain and the heart and the muscles, et cetera, just depending on that gene.
Right. So yeah, because I think a lot of people think that every single nucleotide is a gene. So, like, based on what you're saying, the way to conceptualize MEF2C in particular is if the nucleotides are letters, then these triplet codes make up words, and the MEF2C gene is the paragraph that tells the body how to make that specific protein. Is that a good analogy?
Yes, that is great. We often make the comparison of that our genes are like a big encyclopedia.
Mhm.
So I think that that is perfect. It's a very good illustrative way.
Awesome. Um, can you talk a little bit about exons versus introns? That's something that I've heard talked about before.
Yes, absolutely. So, using the kind of book word paragraph analogy, the exons within a gene is the nucleotides that go to the protein coding material, and introns are the nucleotides that go into the non coding material. So, a gene might have something like we'll just round about for sake of ease. So a gene may have 60 nucleotides within exon number one, and then the intron would be nucleotide number 60 to nucleotide 100, and then the exon number two would be nucleotide 60 to nucleotide 70. So it's only the actual nucleotides within the exons that are going to make that protein eventually. The introns are spliced out. So it's a complex type of, what word am I trying to look for? Not necessarily rearrangement, but a translational difference, if you will, about how the protein is essentially assembled. So, why that's important is because most genetic variants that are going to affect the way that that protein functions are going to be within the exons, because that's the actual nucleotides that are going into making that protein. Now, some people can have genetic variants within the introns, and it's harder to understand the direct impact because that specific spelling change may or may not be going to that final protein structure. But it could be something that's influencing how those exons are spliced together. So even though it's not directly in the meat of the gene per se, it can still have influence on the overall protein.
I see. Another set of terms that gets thrown around a lot is genotype versus phenotype. Can you speak to that a little bit?
Yes, absolutely. So, genotype is referring to the underlying genetic variation. So, something like switching out a nucleotide A for a nucleotide T, whereas phenotype is the physical expression of that underlying genetic variation.
So now we're gonna get into the stuff that I hear people ask about a lot, which is the different
Mhm.
genetic variants. So can you explain the difference between copy number variants and single nucleotide variants, and then we will go into the different kinds of each?
Absolutely. So, going back to the encyclopedia analogy, there can be different genetic variants within our genetic code or our DNA. And something else is that people refer to genetic variants in different ways. I think a long time ago, we used to refer to mutations, as underlying genetic variants or spelling changes. Now we tend to use the word genetic variants. Some people also use the word genetic changes. So I think even that language is changing very rapidly, and even that can be family dependent. Some families may prefer mutation because it's something that it's more concrete almost. But anyway.
I've heard people sort of use mutation as
Mhm,
I can't even completely describe it, but they'll use variation in some contexts and mutation in others. So it is important
mhm,
to know that they're essentially synonyms.
mhm. They are. Yes, they're essentially referring to the same thing. I think people just, prefer different language because it can obviously be very personal when you're
Right.
talking about a child. So do you want to say they have a mutation?
Mm,
It sounds perhaps a little bit harsh, but
right. Yes.
So there are a lot of different types of genetic variants. The most common ones would be what's called single nucleotide variants or SNVs, as well as copy number variants or CNVs. And single nucleotide variants refer to a change that affects a single nucleotide. So typically substituting one nucleotide for a different nucleotide or one letter for a different letter. Whereas copy number variants can involve tens to hundreds to even thousands of nucleotide or letters. So think about much larger deletions or duplications of letters or nucleotides.
Can you explain the different types of single nucleotide variants and sort of how they affect how the gene is read?
Yes. So single nucleotide variants, again, are affecting only one nucleotide. So something that reads within that triplet code ATG, but now we're going to switch out the last G for a C. So now it reads ATC. And one type of single nucleotide variant that would be involving this single nucleotide switch, if you will, is what's called a missense variant. And that means that that single nucleotide switch is causing a different amino acid to be incorporated into the larger protein. Because we talked about the importance of that triplet code, it's very, very specific. So, a specific three sets of nucleotides corresponds for a specific amino acid. So, if you switch out even one of those nucleotides, you're going to make a different amino acid be substituted in place. So, that's a missense variant. A different type of single nucleotide variant would be a nonsense variant. Something that I didn't talk about, but something with that triplet code is that there's a specific triplet code that reads for a stop codon. So this is not corresponding to an amino acid. It's actually corresponding to, okay, we're done making that gene. That gene is done. We are finished. That's the stop codon. So the nonsense variant means that that single nucleotide substitution is not corresponding for an amino acid anymore. It's now corresponding for a stop codon. So you can imagine if a gene is typically 20 exons long, so it has like 20 of these exons that are put together to be made into that larger protein, but somebody has a stop codon in exon number 2, so it's in exon number 2, and now it's saying, okay, stop. Oh, this gene we're done making this gene. Then you produce a much shorter gene than you otherwise would if you did not have that early nonsense variant. So you can kind of imagine the effect of the nonsense variant versus the missense variant, that the majority of the protein and gene is still there. It's just like one little switch out in a very specific spot. Now, of course, if that spot is critical to the way that that protein functions, that may be a bigger issue versus if that exact spot is perhaps not so important. So, missense variants can be really tricky to interpret what they mean. There's a lot of factors that you have to consider versus nonsense variants that are often much more straightforward. A different type of single nucleotide variant would be an insertion or a duplication. So, if we're inserting two nucleotides or 10 nucleotides within a specific point in a gene, that is going to essentially disrupt that triplet code because those nucleotides need to be read perfectly, right in their little sets of three. So if we insert nucleotides, we kind of change how everything after that is read. So those can be, more detrimental to how that gene and how that protein is then read. And that would cause what's called a frame shift variant because we are changing the way that the triplet code is read.
Can you speak to sort of how both copy number variants and single nucleotide variants are sort of read on paper? I know in my son Leo's case, he has uh, a single nucleotide variant, and they show the actual specific nucleotide that's different, and then they show the protein that's different. So maybe you can explain the difference between that as well as the copy number variants.
Absolutely. So, on genetic testing, people will typically get back what is the specific genetic variation or genetic change that was identified from the test. And for single nucleotide variants, how we read that is defined by what's called the C dot and the P dot. So, normally, if we're looking at a lab report, the first thing will be the gene. So, we'll just use MEF2C as an example. And then there's usually a comma, and then C dot or C period. I tend to say C dot, but it's C and then a period. And then there's usually numbers that are designating what nucleotide position we're at. So we'll say we're at nucleotide number 33. And then usually there is the typical nucleotide that's at that position. So we'll just say it's a letter C. And then there's a little caret or a little arrow sign that will point to the new nucleotide that is the new genetic variation. So we'll say 33 C and then the little arrow and then a T. So now the T is the new nucleotide that's incorporated at nucleotide number 33. And then typically there's a little comma and then the P dot, which is going to tell us what is the amino acid change from the nucleotide change. So that usually has the abbreviation for the typical amino acid that's at that spot. So we'll say that's an alanine. There's a three-letter abbreviation for those. So the one for alanine would be ALA. And then 11, which just would designate which number amino acid we're at, and then the new amino acid. So we'll just say that's a valine. So that abbreviation is VAL. So how we write down the single nucleotide variants is the gene, the C dot, which designates the coding change, and then the P dot, which designates the protein change.
Does that Is there any information to be gained from that from parents, or is that something that's more for your genetic counselor to interpret?
Yeah, that's a good question. I think it tells us what is the type of variant. So, like how we were talking about a missense variant, is trickier to interpret just because there's more factors to consider, if you will. Whereas a nonsense variant is designated by with the P dot, it'll have the typical the amino acid position, and then a little star. And that says, okay, this is a nonsense variant, so we would expect that this nucleotide change is going to stop the protein from, or the gene, excuse me, from being created. So that can be important in terms of looking to the future, especially gene therapy options that are exciting. There's other genetic conditions, things like Duchenne muscular dystrophy is the one that comes to my mind because there are available gene therapies for that condition, and they tend to be variant specific. So it's really important to understand the variant to be able to then understand what gene therapies may this person be amenable for. Okay, so How we read the copy number variants is defined by the breakpoints of that deletion or duplication. Since these can often involve hundreds to thousands of nucleotides, usually there is the chromosome position that that gene is involving, something like 16, something like on the 16th chromosome. And then we would define that by which arm of the chromosome and then the specific
So
breakpoints of that deletion or
in MEF2C, that would be 5q14, correct?
Mhm. That
Yes.
is correct.
Okay,
Yes. Yep, and then
gotcha.
parentheses, and then usually a lot of different letters that are defining or excuse me, a lot of different numbers that are defining where does that specific deletion or duplication occur over.
Right, so those are almost like coordinates on a map, just showing
Exactly.
where either something's been put that it shouldn't be or something's been taken.
Exactly. Yep.
Gotcha. Alright, so now moving on to some of those other terms sort of around these variants. You talk about autosomal versus recessive genetic conditions? Or excuse me, autosomal dominant versus recessive conditions.
Yes, absolutely. So, as I talked about before, everybody has two chromosomes, therefore, two sets of genes. And it's important to understand if a genetic variation is on just one copy of the gene or is across both copies of the gene because if a condition is autosomal dominant, then it only takes having one genetic variation across the two copies of the gene to actually have symptoms or features of whatever genetic condition that may be. So something like MEF2C is inherited in an autosomal dominant way. Whereas for autosomal recessive conditions, somebody has to have genetic variants on both copies of that gene to actually be affected with that condition. So, a lot of people are familiar with cystic fibrosis, spinal muscular atrophy, and those conditions are autosomal recessive. So, someone can be a carrier, meaning that they only have one genetic variant and not actually have a recessive condition, they're just a carrier.
So, heterozygous versus homozygous variants, can you go over that as well, please?
Yes. Kind of similar to the autosomal dominant versus autosomal recessive, but heterozygous variants refer to that a genetic variant is only on one copy of someone's two copies, whereas homozygous variants means that it's the same variant on both copies of that person's gene.
Right, and in the case of MCHS, and we still have a lot to learn, so this could change, but pretty much everybody is going to be heterozygous because, as I understand it, what they believe at this point is that if it was homozygous, it would be a non-viable pregnancy and the person would never be born to begin with. And then inherited versus de novo variations.
Yes. So sometimes genetic conditions can be inherited. So a genetic variant can be inherited from somebody's mom or someone's dad, whereas other times genetic conditions or genetic variants can be what we call de novo occurrences, which means of new. So something that wasn't inherited from mom or dad, just a random effect of nature that caused that person to have that new genetic variant.
I think one of the things that a lot of parents are worried about is that because they've had one child with MCHS, that there's a possibility of having another. And from what I understand, this possibility is very small, but it does exist in some people. So, can you speak a little bit about mosaicism, how that works, and how maybe people can test for it if there is a way?
Yes, that's a great question and definitely something that I can understand parents having concerns about. So, when a child is diagnosed with a genetic condition, parents are often tested, whether that be at the same time that the child is originally tested or after, to see if it was something that was inherited from them or was that de novo new occurrence to that child only. And sometimes parents do test negative, meaning that they don't have that same genetic condition, but yet they can still be at risk for having a child that, for having another future child that has the same genetic condition because of this rare phenomenon that's called germline mosaicism. And that refers to a genetic variant being isolated to someone's sex cells only. So for men, be that their sperm, for women, be that their eggs. So when they do genetic testing for a condition or a genetic variant and they're providing something like a cheek swab or a blood draw, that sample is going to test negative or normal because that genetic variant is only in their sex cells. Like you said, it's very, very rare. We estimate that it only happens less than 1% of the time. And that is really a guess because, it's not something that we can ever fully rule out unless you're able to test each of someone's sex cells, to say, can we truly prove does this specific cell that is going to fertilize and produce a child have that genetic variant or not? So, like you said, like I agreed with you, very rare phenomenon, less than 1% is generally what's quoted. But something that people could do would be they could test an embryo before implantation. So that would be going through IVF or in vitro fertilization, or there are other genetic testing options during pregnancy, things like chorionic villus sampling or amniocentesis. And certainly people's doctors can recommend, what may be best for them based on their unique circumstances.
Right. So, in cases of germline mosaicism, not even all of their sex cells may have this variation, right? Is that correct? So it
Right.
might just be 1 or 2 sex cells out of the whole thing. I know there is one family that has multiple children with
Mhm.
MCHS, so it has happened before, but the
Mhm.
chances are extraordinarily low.
They are, and it's hard to get a good grasp in terms of, like, what percentage someone's germline mosaicism may even be,
Mhm.
because the only way to do that would be to sample, each one of their sex cells.
And maybe this is too complicated to give a simple answer, but why does that germline mosaicism actually occur?
Yeah, it's a good question. I don't think that we really know. I think that the theory or the thought is that in that parent, it was a de novo occurrence, but it was something that happened after they were conceived. So something that happened in a cell and then that cell went to become their sex cells, versus like that cell going to become all of their body cells.
I see. So can you speak about the different types of genetic testing?
Yes, there are a lot of different types of genetic tests. So they're all different ways to look at our chromosomes and the underlying nucleotides within the genes that we have. So a test like a karyotype is a very, what I think of as like a 5,000-foot view of the genes and the chromosomes. It's evaluating for the overall structures. So any really large deletions or duplications of chromosomes, something like Down syndrome, that someone has an entire extra chromosome could be identified on a karyotype. Something that is as specific as MEF2C could not be identified on a karyotype, generally speaking. And then a microarray is similar to a karyotype in that it's looking for any deletions or duplications, but it's more specific technology, so it can pick up on smaller deletions or duplications. And then we get into sequencing based genetic testing. So, genetic testing that's actually reading through the nucleotides. And that could be something like a gene panel. So, typically, gene panels are looking at genes related to a specific symptom or diagnosis, something like a hearing loss panel, an epilepsy panel, a neurodevelopmental disorders panel. So, it's a curated list of genes that are being evaluated. And then a much more comprehensive genetic test is something like exome sequencing. So, exome sequencing is looking at the exons or the coding regions of all of the 20,000 genes. Generally, exome sequencing is personalized to each patient that's having it done. So, when a patient is getting it done, whoever is ordering the genetic test and the exome sequencing in this case gives the genetic testing lab clinical notes about that patient's history so that when they're running their analysis, as they're looking across all of the exons over all of the genes, they're paying most close attention to genes related to that patient's diagnoses. So that's exome sequencing. And then genome sequencing is probably the most comprehensive genetic test that we have, and that's looking at exons and introns. So, really looking at all of the genetic material that somebody has. Mhm.
So most people that got diagnosed with MEF2C probably had some sort of gene panel that tested based on symptoms that manifested in the patient, correct?
Mhm.
Yeah.
Yes, I would think so.
Can you talk a little bit about how, when someone actually gets a genetic test, how they go about interpreting their results and looking at that piece of paper and actually making some sense out of it?
Yeah, I think that is the $1,000,000 question. And genetic lab reports have huge variability. Some are written for clinicians that they are assuming are going to do the bulk of the interpretation. But now I think with the advent of a lot of my charts and electronic health records, parents are getting genetic test results. Perhaps earlier than their doctors that ordered it. And so they're not always written in a very patient-friendly way and can be, very bulky, especially with different terminology and things. So I think if families are wanting help interpreting their genetic testing results, reaching out to the clinician that ordered it can be really helpful. But then there's also resources like us, like Simons Searchlight, that, we have a team of people available that are willing to sit down with folks and really go through things to help them understand.
Yeah, that's great. Yeah, and it seems like, there's only, unless you are a trained geneticist, there's only so much information to be gleaned from that report. I got our report. I could look at it and sort of intuitively go, I don't remember what the nucleotides were, but
Mhm,
I could look at it and kind of, based on the context, be go, oh, he has a, a this where he should have a that. But then everything else
mhm.
is just too over my head. And unless you are a trained geneticist, just don't don't bend yourself out of shape trying to figure this stuff out. Is
Yeah.
the advice I would give people.
It's really like its own language. You have to have all of these basics to even begin to interpret it. And even, for people that have been in this world for a long time, it's ever evolving. So it's hard.
So we're going to move into some MEF2C specific questions here. Do you know what different types of genetic variations are seen in MEF2C? I know, again, data is extremely limited, but if there are any very broad generalizations that you might be able to make based on our very limited data.
Mhm. Yeah, so there is a gene reviews for MEF2C that was published and updated in December of 2024. So relatively recent based on the published literature that's available about MEF2C. And the majority of patients that have been identified have a single nucleotide variant. So whether that be a missense variant or a nonsense variant or a frameshift variant, that seems to be anywhere from 65 to 70% of patients that fall within that bucket. And then the remaining 30 to 35% of patients have a copy number variant, so some larger deletion or duplication that involves MEF2C.
Just a quick little self promotion here. The main two authors on that gene review, are Dr. Skinner and Dr. Cooley Coleman. And we're going to be interviewing both of them in July. So hopefully we'll be able to get some more insight on that for our listeners. But um, do there seem to be any patterns between specific variations and, I guess the technical word would be phenotype, sort of manifested symptoms?
Yeah, I think what you're referring to is what we call genotype phenotype correlations, which have been identified for some genetic conditions that we have a lot of data on, that we can have 500 people that are within this genotype that are then presenting with this phenotype, and then, another few hundred people that are with this genotype presenting with this phenotype. I think at this point for MF2C, we don't have quite the numbers to be able to make any conclusions. So I think that that certainly speaks to the importance of participating in research projects to be able to pull data, to be able to answer some of these questions that families have.
Sure, absolutely. So, should parents consider regular retesting as our genetic technology advances?
Yeah, I think that this is a really good question and definitely something that families ask for a lot. I think with genetics, and we're at a point with our genetic testing technology, like our ability to sequence genes, we think is pretty good. So it's not necessarily that we expect somebody's genes and their nucleotides to change over time, but what changes and is really meaningful is our ability to interpret what the significance of genetic variations are. So I think that's the value of rerunning genetic testing and seeing, okay, is there any new technology or any new discoveries that we can make within how we can interpret mine or my child's genetic code that we couldn't when they were originally tested, several years ago. So that's certainly something that doctors and genetic counselors can, look at families' previous testing to say, okay, does this warrant reevaluation or does it not?
Your medical professional would be able to recommend sort of a regular interval of testing if that's necessary and which tests to run as well.
Absolutely.
Yeah, perfect. Alrighty, so how can the listeners get involved with Simon Searchlight and why should they get involved with Simon Searchlight?
Yes, I think there are so many reasons to get involved with Simon Searchlight. If I may just give some brief background,
Yeah, go ahead.
Simons Searchlight accelerates research into rare genetic neurodevelopmental disorders, including the MEF2C gene. And we do this by collecting high quality natural history data and also building partnerships with families, researchers, and advocacy groups like MEF2C, because we're all very invested in trying to move the research forward. So, how we actually do this with patients is we collect online research surveys. There's also the opportunity to provide a blood sample. This isn't mandatory, it's an option. And through the online research surveys, we're gathering detailed medical and behavioral information. And then we make all of that data available to researchers in a de identified way. We're very cognizant of this because we want to protect patients' privacy. That is very important so that there's no way to track down, who is this specific patient. We're very careful about privacy. And like I said, we share that data with researchers, but then we also give it back to families. So on our website, we have things like gene guides, quarterly reports, other support resources, so that it's not just you feeding data into us. We're actually feeding that data back into you to inform anything that you may want to know. Participation in Simon Searchlight is free, and in fact, families are earning gift cards for completing their surveys because we know that your time is so valuable.
Yes,
Um,
we unfortunately, because we're special needs parents, we never have time to use them, but we have several gift cards for date nights at the ready.
yeah, it's free. We give a little bit back for your time because we
Yeah.
do appreciate it, a little
Mhm.
incentive.
What I would add is that, at this stage with MEF2C being as underdiagnosed as it is, I've been told there's some unpublished data that estimates about 1 in 30,000 people have a MEF2C variation. There are other estimates. And so, um, you know, again, we don't know much. But my point is that participating in Simon Searchlight helps us know more, and uh, ugh, that will eventually lead to better treatment and uh, also, Dr. Chung on our episode talked about diseases that are too rare to care. So we can avoid that situation by getting as much good data as possible on MCHS. So those are other reasons why it's important to participate in things like Simon Searchlight if you have the opportunity.
And I think from a parent's perspective, you want to be represented. You want your child to be represented and their data to go to the larger understanding to hopefully inform those families that are coming behind you. So it is some of that like altruism as well that can really help with research participation.
Right, absolutely. Yeah, we've talked
Mhm.
about that on the show before because, like, let's say, I'm not very good at math, but let's say that 1 in 30,000 number is correct. As genetic testing becomes more commonplace, more
Mhm.
people are diagnosed, your data could literally help, hundreds, thousands of people over the course of generations. So it's actually really exciting and empowering thing to think about, in my opinion. But is there anything else you'd like to share with the listeners?
I think find your community. The importance of community is something that families hold so near and dear. And I'm obviously preaching to the choir here, but just the importance about building family bonds and making connections with other parents is so important. The importance about participating in research, which we talked about to really inform clinical trials or future therapeutics, and just really moving the needle forward in a meaningful way is so impactful. So just try to stay engaged.
Yeah, absolutely.
Mhm.
Alrighty, Jamie. Well, thank you so much for your time. It was really great to have you. I think a lot of people are going to find this really valuable. So,
Of course.
as I mentioned before, we have lots of great episodes coming up. You're the last interview in our we did 6 in 6 days. So,
Oh
um,
my gosh, that's
we're very busy.
crazy.
Yeah, and we
Yeah.
hope to have lots of great stuff out to people soon. So,
Mhm.
as always, they can find us on Spotify, Apple Podcasts. Contact us on our Facebook page and email us at mef2cast@gmail.com. So thank you to everybody listening.